I'm Corey.
I'm Anthony.
And I'm Andre. And welcome to Crash Course Biology, Cellular Transport.
Let's talk about the cell membrane. It acts as a shield or a blanket that surrounds and protects the entire shell. It's made up of two layers of phospholipids, which they call it a phospholipid bilayer. The phosphate head is hydrophilic, which means that it loves water, and it's polar. There are two hydrophobic fatty acid tails that are nonpolar, which means they hate water. Since a large portion of the plasma membrane is hydrophobic, then molecules that are small and nonpolar can easily pass through. Besides the phospholipids, there are other lipids in the plasma membrane. First off, there's the glycolipids. Just like the phospholipid, they have two tails, but instead of a phosphate head, they're connected to a chain of carbon sugars. The other lipid is called cholesterol. It has a rigid structure that sits near the tails and makes that portion of the plasma membrane slightly more firm. This decreases the permeability in that region and makes it so that only specific molecules can pass through that region. Just like the different tiles that make up a mosaic, there are lots of different proteins that make up the plasma membrane too. These proteins are embedded either partially or wholly into the plasma membrane. One type is the peripheral proteins, and they're found on the outside surface or on the inside surface of the cell. Integral proteins are found embedded within the membrane. That means that the hydrophilic regions poke out just like the phosphate heads. There are other integral proteins like glycoproteins, and these proteins, just like the glycolipids, have a carbon chain that reaches outside of the cell. The cell membrane has a very fluid consistency, just like that of oil. That means that the phospholipids can slide across each other and move across their layer. It's very rare that they move between layers. Proteins can also move across if they're not connected to the cytoskeleton. All of the cell membranes are made out of different kinds of proteins. Some integral proteins help to transport materials that can't just pass through the cell membrane. These are the channel proteins and the carrier proteins. Other proteins are receptor proteins, and they have specific molecules that bind to them on the outside and can cause a response in the cell. Other proteins are enzymatic proteins, and they catalyze reactions so that they're easier to occur.
Let's learn about diffusion. Diffusion is the movement of gases in a cell from high to low concentration until the concentration is equal on both sides of the plasma membrane. Oxygen and carbon dioxide are lipid soluble so that they can move across the plasma membrane very easily. Osmosis is the diffusion of water into and out of the cell. In this scenario, water is the solvent. A solvent is what the solute is dissolved in, and the solute is the substance that is dissolved. Isotonic solutions are where the concentration of water in the solution is the same on both sides of the plasma membrane, so there is no net movement of water. Hypotonic solutions are where there is a lesser concentration of solute outside the cell than in the cell. When a cell is placed in a hypotonic solution, water enters the cell and it swells. Hypertonic solutions are where there's a greater concentration of solute outside the cell than in the cell. When a cell is placed in a hypertonic solution, water exits the cell and it shrivels. One example is plasmolysis, which is the shrinking of cytoplasm due to osmosis. This occurs when cells are placed in hypertonic solutions. Facilitated diffusion is the transport of substances along their concentration gradient, requiring the use of a carrier protein. This works like the airlock on a spaceship. The astronaut comes in from space, the door opens, and then the door seals. This allows for the pressure to equalize, and then the door to the spaceship opens and the astronaut exits. Carrier proteins are specific to a single molecule, and carrier proteins are also reusable. Since glucose and amino acids are larger molecules, they undergo facilitated diffusion and therefore need the aid of a carrier protein in order to get across the plasma membrane.
Now let's learn about active transport. Small molecules like oxygen and carbon dioxide that are hydrophobic and nonpolar are able to pass through the membrane through something called passive diffusion, which requires no energy. When it goes with its concentration gradient from high to low concentration, no ATP or energy is needed, meaning that this is a passive transport, almost like you're pushing something downhill. Active transport is when a molecule is going against its concentration gradient, and in order for this process to work, ATP is needed. So it's like if you're pushing a rock up a hill, you need energy in order to push this rock up the hill. Ions and polar molecules, which are charged, can't enter the cell membrane like oxygen or carbon dioxide because they need something called the channel protein in order to pass through the cell membrane. They pass through the channel protein by having the inside of the channel protein actually conform to the same charge and allowing it to pass through it. Another way that something can enter the cell is through a process called endocytosis, which is usually in regard to bacteria. Take, for example, a white blood cell, which will see a foreign substance inside the body and will eventually have to take down the bacteria. In order for it to destroy the bacteria, it needs to go through endocytosis. Endocytosis requires organelles inside the cell, including the plasma membrane, the Golgi apparatus, the vesicle, and the lysosomes, which all have specific jobs. Basically, what endocytosis does, it conforms around the bacteria in something called a vesicle, which is important in transporting the bacteria throughout the cell. Once it goes out of the vesicle, it goes into something called the Golgi apparatus, where it modifies the bacteria in order for it to be broken down. After the Golgi apparatus, it then goes into another vesicle where it is carried to something called the lysosome, where the bacteria is completely broken down and killed. On the other hand, there's another process called exocytosis, which is the inverse of endocytosis in that it brings things outside of the cell. Exocytosis uses the endoplasmic reticulum, the vesicles, the Golgi apparatus, and the plasma membrane in order for it to function. Exocytosis starts inside the rough endoplasmic reticulum, which synthesizes proteins. From there, the protein is taken into something called the vesicle, which we talked about before. While inside the vesicle, the protein is transported into the Golgi apparatus. From the Golgi apparatus, the protein is modified and it is taken into the vesicle, and it is transported outside of the cell into the body. The sodium-potassium pump allows sodium and potassium ions to go through the plasma membrane and is really important in nerve cells. The two pump in different directions, and in doing this, they create a gradient, which is important in allowing other processes to happen that we have talked about earlier in the video. Sodium requires energy in order to flow outside of the cell, and 3 molecules will exit the cell in the process. Potassium, on the other hand, will flow inside of the cell and requires no energy at all. Sodium and potassium originally start on the opposite sides of the cell. The sodium starts on the inside of the cell and the potassium starts on the outside of the cell. In order for the sodium to be pumped out of side of the cell, you need the channel proteins, which we talked about earlier in the video. For it to work, only 3 sodium ions will be pumped outside of the cell. So the 3 sodium ions will go into the channel protein, and ATP will be required in order to push this through the membrane. We talked about this earlier in the video, and it is known as active transport. So sodium requires active transport in order for it to be pumped outside of the cell. Once this happens, the potassium ions will go into the channel protein, and only 2 of them will be required to go inside of the cell. For this to happen, absolutely no ATP is required. So it is similar to what we talked about earlier in the video, and that is the passive transport. So the potassium will go inside the cell, and the sodium will be outside of the cell after the pump has been done. And this creates the concentration gradient.
Thanks for watching. Yep.
Water lays the cell. Oh.
They're connected to two.
Phospholipid head. Alright, there.
Wait, so you have a tripod valve, right?
2 layers of phospholipids.
Christian Shoshana to the West Office, Christian Shoshana to the main office.
Let's talk about diffusion.
Good.